In this video, we will learn about digestive glands. So first, we will see salivary gland. Three pairs of salivary glands open into the buccal cavity. Parotid glands located just in front and beneath the ears. So this is parotid gland. Then submaxillary glands located close to the inner side of the lower jaw, one on either side. So this is submaxillary gland. Then sublingual glands lying below the tongue. So this is sublingual gland. The salivary glands secrete saliva. Saliva is viscous, transparent, slightly alkaline liquid with pH equal to 7.2. It contains mainly water, salt, and mucin. Functions of saliva. It lubricates and moistens the food and binds the food particles into a bolus for easy swallowing. The enzyme salivary amylase present in saliva converts starch into maltose. Saliva is antiseptic. It contains enzyme lysozyme, which kills germs present in food and in mouth. Then liver. Liver is the largest gland in the body. It is reddish brown and weighs about 1.5 kg. It lies on the right side of the abdominal cavity in the concavity of diaphragm. So here this is diaphragm and this is liver. It is formed of hepatic cells. Liver produces bile juice which is stored in gallbladder. So this is gallbladder and is released in duodenum when required. So this is duodenum. Gallbladder is transparent, ovoid sac embedded in one of the liver lobes. It receives bile from the liver via hepatic duct and stores it. The gallbladder opens into duodenum through common bile duct. So this is common bile duct. Then pancreas. Pancreas is diffused pink-colored leaf-shaped gland. This is pancreas. It lies in the U-shaped loop of duodenum. Pancreas is a dual gland. Its exocrine part is formed of acini or pancreatic lobules. The acini produce pancreatic juice which contains enzymes for the digestion of sugars and proteins. The endocrine part of pancreas is formed of islets of Langerhans. These produce insulin and glucagon hormones which regulate glucose level by decreasing and increasing its amount in blood respectively. The pancreatic duct releases pancreatic juice in the duodenum. So this is pancreatic duct. Then digestion. Digestion is a process of physical and chemical breakdown of large and complex molecules of food into simple and small molecules by the action of digestive enzymes so that they can diffuse into blood through the gut wall. First, physical digestion. So this is chewing and mastication of food so that digestive enzymes can work on them. Then chemical digestion is breaking of nutrient molecules of food by the action of digestive enzymes. Then digestion in the various parts of alimentary canal. First, digestion in buccal cavity. The ingested food is chewed and masticated in buccal cavity. Mucus in saliva moistens and lubricates the food, makes it soft so that it is easily swallowed. Salivary amylase present in saliva breaks starch into maltose. Then pharynx and esophagus. No mechanical or chemical digestion occurs in pharynx and esophagus. They just pass the food from buccal cavity to stomach. Then digestion in stomach. Protein digestion begins in stomach. The muscular wall of stomach undergoes alternate contractions and relaxations and churns the food into a fine pulpy mass called chyme. Gastric juice contains hydrochloric acid, digestive enzymes, propepsin, prorenin, and gastric lipase, and mucus, that is gastric mucin. Functions of hydrochloric acid. It makes pH of food from alkaline to acidic. That means pH is 1.8. It destroys bacteria present in the food. It activates the inactive propepsin into active pepsin and prorenin into renin. It also controls opening and closing of pyrolytic aperture. So we will see the action of pepsin. It breaks or hydrolyzes the protein molecules into peptones and proteoses. So propepsin, that is in inactive form with the help of HCl, first gets converted into pepsin, that is in active form. Now this pepsin breaks down protein into peptones and proteoses. Then renin. In the presence of HCl and calcium ions, renin helps in the digestion of milk protein, that is casein. So first, inactive form, that is prorenin, with the help of HCl gets converted into renin, that is the active form. Now, with the help of renin, this casein or milk protein is converted into paracasein. Now, this paracasein combines with calcium ions to form calcium paracasinate, that is insoluble. Now, this calcium paracasinate with the help of pepsin breaks down into proteases, peptones, and polypeptides. Then gastric lipase, it partially hydrolyzes fats into fatty acids in glycerol. Then gastric mucin, it protects the mucous membrane of stomach. Then digestion in small intestine. In duodenum, the partially digested food or chyme from the stomach is pushed into the duodenum by the relaxation of pyrolytic sphincter. The bile juice from the gallbladder and the pancreatic juice from pancreas are poured into duodenum and mixed with chyme. Then, function of bile juice. Bile juice has no digestive enzymes, but it is important for the digestion because bile neutralizes acidity of the chyme in the duodenum. It makes the medium alkaline, so pH will be 8.0 to 8.6 for the action of enzymes of pancreatic and intestinal juice. Then bile emulsifies fats and prepares them for the action of enzymes. Then the role of pancreatic juice. Pancreatic juice contains following enzymes: trypsin and chymotrypsin. Both trypsin and chymotrypsin are secreted in inactive form called trypsinogen and chymotrypsinogen. On being changed to active form, they bring about protein digestion into polypeptides. Then carboxypeptidase, it hydrolyzes polypeptide chains into tripeptides, dipeptides, and amino acids. Then pancreatic amylase or amylopepsin. It hydrolyzes the remaining starch and glycogen into maltose, that is a disaccharide. Then pancreatic lipase or strypsin. It acts on emulsified fats and hydrolyzes them into fatty acids and glycerol. Then pancreatic nucleases. Ribonuclease, that is RNase, and deoxyribonuclease, that is DNase, hydrolyze nucleic acids, RNA and DNA respectively, into nucleotides and nucleosides. Then, in ileum, the intestinal glands in ileum secrete intestinal juice, that is succus entericus. It acts on the end products of digestion completed in duodenum by pancreatic enzymes and completes the process of digestion. Succus entericus contains the following enzymes. First, pepsin or peptidase hydrolyzes polypeptides, tripeptides, and dipeptides into amino acids. Then maltase hydrolyzes maltose, that is a disaccharide, into glucose, that is a monosaccharide. Then sucrase or invertase hydrolyzes sucrose into monosaccharides, that is glucose and fructose. Then lactase hydrolyzes lactose into glucose and galactose. These are also monosaccharides. Intestinal lipase emulsifies the remaining fat into fatty acids and glycerol. Enterokinase converts inactive trypsinogen into active trypsin enzyme. Nucleotidase or nucleosidase hydrolyze nucleotides and nucleosides respectively into nitrogenous bases, pentose and hexose sugars, and phosphates. Then digestion in large intestine. Water and salts are absorbed by the mucous lining of large intestine and undigested residue is changed to semi-solid phases. These are pushed into rectum from where they are passed out through anus by the process of egestion or defecation. I hope this topic is clear to you. Thank you.